introduction to inbreeding species yesterday, and today we'll continue with an introduction to outbreeding species. Thank you, David.
Okay, thanks, Catherine. For those who weren't present yesterday, I'm David Lloyd. I'm head of forage breeding for Germinal. And yes, I'm going to be talking about the breeding methodologies you would use traditionally for outbreeding species. So I covered inbreeding species yesterday. This is where individual genotypes within a population tend to self-pollinate before they can be outcrossed. So breeding methodologies used within breeding species tend to focus on producing pure line cultivars, where individual genotypes within the population share a high level of homozygosity with other members of the population. Now, this approach isn't suitable for outbreeding species. They tend to have robust self-compatibility mechanisms, so they naturally outcross, and these self-incompatibility mechanisms can be genetic, morphologic, or phenologic for that matter. And populations tend to have a high level of heterozygosity. And we want to maintain this level of heterozygosity to some extent because outbreeding species tend to be susceptible to inbreeding depression. They carry a high load of deleterious recessive alleles. The approaches used for variety development tend to use population improvement. So we produce open pollinated population cultivars. These harness the heterozygosity, and we focus on production of sufficiently genetically distinct but phenotypically similar genotypes within the breeding population. And a valid comparison for this is with animal breeds, where each individual genotype is distinct from other members of that breed, but they are all sort of phenotypically similar. So open pollinated cultivars still have to adhere to DUS requirements: distinctiveness, uniformity, and stability. There's more scope for a population cultivar to fail the uniformity element of DUS. So sometimes the requirements are somewhat looser. This is a double-edged sword. You tend to meet the requirements for uniformity more often, but there's more of a possibility for failing the distinctiveness aspect of DUS. Open pollination is common in forage legumes, such as clovers, red clover there on the right, and alfalfa. There's some open pollination with some grain legumes. So faba bean can be treated either as a pure line cultivar or open pollinated cultivars. In practice, combinations of the two approaches are used for faba bean. Pedigree methods often used to produce inbred lines that are subsequently combined as open pollinated synthetic varieties. So the breeding objectives we have for outbreeding species are similar to those for inbreeding species. Yield and potential yield is often the largest concern. With forage legumes, there's a difference, however. We're looking at the above ground vegetative parts of the plant, so-called herbage yield. And with perennial species, we're often looking at multiple cuts over multiple years rather than a single harvest. Yield stability is important, so we're quite concerned about the response to abiotic stress and also for biotic stress. And in the biotic stresses include things like grazing tolerance. While it's not the major objective of red clover and alfalfa, these are being looked at by various groups. It's important in other species such as white clover. Resistance to diseases is also important. So-called clover sickness is important with red clover. This is a combination of Sclerotinia crown rot and Duttonellus nematodes, stem nematode. Anthracnose and bacterial wilt are major pathogens of alfalfa. These are the major ones in the UK at least. But the importance of various diseases varies in different environments. Quality traits are important considerations. So with forage crops, digestibility, fiber content, and protein content are important. In red clover, Isoflavones, so-called phytoestrogens, are a very important consideration. These can have estrogenic effects on livestock, causing fertility problems. And persistency is a major goal of breeding with forage crops. So this is yield stability in response to cutting multiple times over multiple seasons. So sometimes initially high-yielding varieties can have a tendency to burn bright and fast and suffer persistency issues later on in their life cycle. There is a temptation to concentrate on herbage yield and treat seed yield as an afterthought. Now, this is a mistake, because it doesn't matter how well a variety does in terms of its agronomic performance. If you can't produce sufficient seed, it will not make it in the commercial sector. So founder populations, the origins of a breeding population. The first step in outbreeding populations is to create or identify a sufficiently diverse breeding population. This can be a pre-existing population, such as ecotype selections involving collections of wild material, or it can be refinement of existing varieties. There's a lot of old varieties out there that have come off national lists and have very promising qualities that can be harnessed, but may fail to meet other aspects of modern varieties, such as the uniformity requirement for DUS characteristics. Of course, if you're using existing varieties, care is needed to avoid DUS issues, particularly with the distinctiveness aspect. So if it's a fool's errand to try to refine an existing variety that is still current. Arguably, a better approach is to take divergent material and hybridize it, similar to the approach used in inbreeding populations. So hand crosses can be used effectively, but flowers are generally a lot smaller than they are in grain legumes. It's very difficult to do. You need good eyes and a steady hand, and I'm afraid I don't have either of those. And you need to cross sufficient numbers to avoid inbreeding depression. So you need to be successful a number of times and to be able to produce a valid breeding population. Insect pollinators can be used effectively, although they're not very discerning about what they pollinate. Top crossing approach can be very effective. So in the first picture there on the right of the screen, there's one plant of population A in the middle of an insect-proof isolation chamber, surrounded by many plants of population B. Insects are used to pollinate freely there, and you rely on self-incompatibility to prevent selfing within plant A in the middle. Seed is only harvested from the mother plant, from the plant in the center of the insect-proof chamber, and all seed collected will theoretically be an F1 cross between the two populations. And we tend to combine balanced bulks of multiple reciprocal top crosses in subsequent polycrosses to sort alleles into Hardy-Weinberg equilibrium. Space plants, they have their drawbacks. They don't perfectly imitate plant behavior as it exists in a lay or stand. It's still the most efficient way to phenotype at a large scale, though. Breeding's a numbers game. If you want to select sufficient plants at high enough selection intensity to achieve good genetic gain, you need to start with and characterize a lot of plants. So if you need a minimum of 30 plants, which is a realistic figure to maintain heterozygosity within a population, and you want to impose 1% selection intensity, you need to start with at least 3,000 plants. It's inefficient to do this with closely packed plants. And it's also worth mentioning that most DUS testing is done on space plants. So there's a valid reason to select for that under conditions where the plants are actually tested. In red clover, we tend to do this by growing plants in pots. Start with perhaps 2 or 3 times the number that we intend to put out in the field, They're assessed within the glasshouse for various traits: cotyledon size, leaf color, markings. Petiole hairiness is quite an important trait in red clover. We'll look at the general vigor of the plant in the pot and remove any outliers. We then establish the plants within the field in springtime. And in the establishment year, we look at various traits, things like plant size, vigor, and its tendency to flower without vernalization. Some plants will flower, some plants won't. In the second year, we tend to collect a lot more data. This is following its first winter. Look at the growth habit, whether the plants are erect or prostrate, flowering dates, leaf color, leaf size and shape, plant sizes and vigor, height, stem thickness, etc., etc. The art is in selecting subpopulations that combine sufficient uniformity with performance. It's sometimes possible to select several subpopulations with, for example, different flowering times. The selected plants are then polycrossed to produce the next generation, which polycrossing involves putting plants into an insect-proof glasshouse and allowing insects to freely pollinate all plants. It's worth mentioning mass selection. This is one of the oldest forms of selection used within plant breeding. Essentially uses the same principles as natural selection. A breeding population is subjected by a stress, and this stress is controlled by the breeder, theoretically. It can be abiotic. It could be putting plants into a lower soil pH than is optimal for them, or a low nutrient content. We've used this approach very extensively to select, for example, for phosphorus use efficiency in white clover. It can be biotic stresses. This is disease and pest load, grazing intensity. We've used intensive grazing to select for grazing tolerant red clovers, for example. And on the right, you can see one of our imposed biotic stresses. He's probably been exposed to some biotic stress of his own and been eaten by now. After a certain length of time, survivor plants are then selected and polycrossed. It's an excellent way of selecting for specific environments, but it is very difficult to control precisely. You have to balance the challenge of the environment you place the plants into. If it's too minor, you don't get insufficient selection intensity. If it's too harsh, you tend to kill all the plants. Recurrent selection. This is a commonly used approach for population improvement. Used conventional selection methods, whether it's mass selection or phenotypic selection. Select the best plants from the breeding population, polycross the best plants to form an improved population, select again from that improved population, and repeat ad infinitum. A balance needs to be made between genetic gain and maintaining genetic diversity within the populations. If you use a harsh selection intensity, you tend to get the best genetic gain, but you tend to lose genetic diversity, and improvement to the population in subsequent generations can plateau. So in practice, it's best to select two populations, one that has had a harsher selection intensity, which you use for variety development, and a larger selection of plants that has less stringent selection intensity for population improvement. Typically, the smaller population will be a subset of the larger population. Progeny selection, sometimes crossing genotypes that look the best, that have the best phenotypes, is not the most effective approach. We call this crossing the best with the best and hoping for the best. Visual appearance does not guarantee how well a group of plants will combine in a cross. So we use an approach called progeny testing, using half sieve families. It's possible to use full sieve families as well, but I'm going to cover just half sieve families. This is the approach I'm more familiar with. In this approach, a breeding population is polycrossed and the mother plants are retained. This obviously applies for perennial plants. It's difficult to retain the mother plants of annual crops, but the approach can be adapted for annuals. Seed from the mother plant is harvested separately from other mother plants and retained as what we call half sieve families. This is where each individual of that family is a half sibling. They all share the same mother plant, but have different pollen donors, or father plants, if you like. Those half-sib families are then sown in trial plots, and the best performing half-sib families are identified. The assumption is made that the best performing half-sib families are from the mother plants that combine the best. They've got the best combination ability with other genotypes. At this point, we go back to the identified elite mother plants and polycross them to produce a new variety. And on the right there, this is a diagram I believe was produced initially by Leif Skutt. It's the approach used at IBERS and by Germinal for forage breeding. On the outside circle is the recurrent selection population development stage, and on the inside is the half-sib family selection, which is used for the variety development. This has been a very effective approach, and it has been used for the last 30 or 40 years by IBERS. So that's in a nutshell how we breed open pollinated outbreeding crops. Obviously, this doesn't cover the entire subject. We'd be here for some days if I tried to do that. There's a number of things I haven't covered. For example, ploidy doubling to induce tetraploidy in red clover, or interspecific crosses. I'm happy to talk about these if there's any specific questions about them, or indeed to answer any questions about the rest of the talk. Thank you.
Thank you very much, David. That was very comprehensive. At the moment, we have no questions for you. You'll be glad to hear. But I think many of the questions will probably come up in the individual species talks, which you provided an excellent introduction to. And we now have a slight break, so to enable people to get a cup of coffee. I'm afraid we can't offer you coffee or opportunity to chat with the speakers this morning, but you'll have to do that for yourselves. But we'll resume at 10:50 for the next two talks, and they will be given by our two species experts on alfalfa and on red clover, which will follow very nicely from the introduction that David has just provided on the breeding systems for those crops. So thank you very much, and see you again in about